So we talked about where spectrophotometry can be used in your assays. Number one, color-based concentration assay. Number two, turbidity-based quantification assay, especially when you are quantifying the amount of cells present in your growth medium. Number three is the kinetic assay, when you can measure the speed of enzymatic reaction. So spectrophotometer can be used. Okay, and whatever the assay you're going to use, your spectrophotometry, these are all important suggestions. Number one, sample should have the detectable range of analyte concentration and should not contain interfering substances. What does that mean? Detectable range means what? You should not have too little or too much of the analytes. So you should have, the detectable range that shows the linear relationship. Okay, and then anything that bothers the light observance should not be there, like any bubbles or any aggregates or anything that hinders the light measurement should not be there. If you find something like that, you have to get rid of it before you assay. And then choosing the proper wavelength for your measurement is very important. Okay, so make sure you are using the correct wavelength that observes your analytes very well. So if you don't know what that is, you may have to do the scanning spectroscopy and see at which wavelength your light is best observed on your sample. And then validation of a method should be done considering the range of an assay, like I said. Okay, the lowest amount of your analyte, the highest amount of your analyte, in between the threshold you should use your assay. And sensitivity, again, change the observance versus change in analyte concentration. So the threshold, again, it's the issue. Okay, if you're above the threshold, you cannot use that observance in your estimate concentration assay. Selectivity, the ability of an assay to distinguish the compound of interest in the presence of other material. If it's a copper, what is the observance it is detecting copper? Well, if it's some other metal, what is it? You have to choose the correct one. There should not be any overlapping observance that hinders the measurement, right? And the ruggedness, the degree of the reproducibility of a test result obtained by the analysis of a sample under variable, normal test condition. Okay, so it should be reproducible, of course. And the accuracy, the closeness of the test result to the true value should be considered, and precision as well, long-term reproducibility. All these things are very important. Okay, and then protein assay, going back to that, we talked about Bradford assay using the Comasi brilliant dye molecule G250 that sticks well with the protein and it observes the blue light very well. But besides Bradford assay, there are two different kinds, like BIOT assay or Lowry assay. Okay, BIOT looks like this. It has copper in it, so it gives kind of a reddish color. And Lowry will give it another color as well. So you can use any of these three, but they have different characteristics. They have different range they can cover. They have different reactivity with different solution. So considering all those, you need to choose the best assay for your measurement. And among those, I think Bradford assay is the best protein assay because it's I guess sensitivity is good, and it also covers the wide range and things like that. Okay, so I may show you Bradford assay versus Lowry assay, the difference. So Bradford assay and which one is the Lowry assay, the double line? I guess Bradford assay is the right, I guess it's the outside. This one, and Lowry assay maybe another one. So anyway, consider the difference where they are covering and the accuracy as well. And then you can use the UV observance in quantifying amount of DNA, RNA. Hopefully you will do this lab, or you already have done the lab, okay, whichever the case, So UV260, okay, is used especially when you try to detect the amount of DNA, RNA, nucleic acid. Okay, especially between the concentration of 5 and 100 microgram per milliliter range, that linear relationship exists between the observance and the concentration. Okay, so based on that correlation, you have the reference point. Okay, at 260 nanometer UV light, double-strand DNA, it absorbs the light very well, especially if that light absorbance comes out to be 1. Normally you have that much of amount of DNA, 50 microgram. Okay, so using the reference point, you can calculate or estimate the amount of DNA present in unknown sample. Let's say your unknown sample has 0.5 OD, then how much of DNA will be there from 260? Then it's about half of the concentration, 25 microgram per mil. That's straightforward, right? And then if your sample is made up with single strand DNA instead of double strand, the observance reference point is a little different. 1 OD equals 33 microgram per mil. Okay, because single strand DNA tend to absorb more light, and the pure RNA absorbs more light than even the single strand DNA. So 1 OD, I guess it's in between single strand and double strand DNA. So it's 1 OD equals about 40 microgram per mil of single strand RNA. So you have to use that reference point in RNA concentration estimation. Okay. Also, the purity of the DNA, RNA, protein samples can be estimated using the ratio. But before that, we're not using the UV light absorbance in protein as quantification. I told you the reason why, because proteins have different light absorption profile based on what amino acid they have. But if you have to use it, then you have to use it instead of 260, you use 280, and you assume that OD of 0.7 if you're using that absorbance for like a BSA protein calculation, and 0.7 equals 1 milligram per mil. Okay, that reference point should be used for what? Only for BSA sample, not other proteins. So you have to understand the reference point for each protein would be different. Okay, and if it's antibody, it's about that observance that equals about 1 milligram per mil. So since it's not really accurate, you're now using this UV observance for quantifying protein samples. Instead, you use Bradford assay. And then the ratio, like I said, 260 over 280. And that gives you the purity of RNA sample or DNA sample double-stranded or DNA samples with protein. Okay, because if that ratio comes to be close to 2, that means you have a bunch of RNA, mostly RNA only. If the ratio happens to be close to 1.8, that means you have mostly DNA molecule only. If the ratio happens to be very low, about 0.6, that means you have a bunch of proteins in your sample. Okay, by looking at the ratio, you can tell how pure your sample is. Okay, so that's the end of the lecture 5.